All right, guys. In chapter 24, we're going to get into um, the origin of species. In other words, we're going to talk in this chapter about how new species are formed. As we saw in some of the previous chapters and discussed with Darwin, he saw crazy stuff on the Galapagos Islands that he didn't see anywhere else on Earth, right? And throughout the material we've touched on with evolution, you've been exposed to new animals and new behaviors and crazy stuff that you never really would have thought of either. And so when we see these new things, it all speaks to the origin of new species. The idea that Darwin was trying to get across here was that all of the diversity that we see in our world, the new species that we see, the diverse species that we see, was brought about by this theory of evolution. So evolutionary theory must explain how new species originated and also how populations evolved. Specifically, one of the ones we've yet to talk about I should go back here. Chapter 23, we talked a lot about microevolution, or microevolutionary change at the population level, these small allelic changes from generation to generation could lead to variation and differentiation in species. And the adaptations and traits that we see within the species, whereas macroevolution refers to this evolutionary change above the species level. In other words, big changes, taking them, I've mentioned this a couple times, monkeys to humans or dinosaurs to birds, things of that nature. And so we've really yet to kind of talk about how that would ultimately occur. So that's what we're going to do here in this chapter, is start to discuss some of the factors that are surrounding this large macro-scale evolutionary change. Now, there's two basic theories of how this macroevolutionary change occurs, one called anagenesis and one called cladogenesis. These are more easily understood when we look at a diagram, a picture. But anagenesis is the conversion of an entire species to a morphologically different form, generating a new species. In other words, this would be a direct line of evolution from, for instance, great apes all the way to humans with no branches whatsoever in that pathway. The evolutionary line does not branch at all. There's no increase in species diversity. It basically says there was an ape, and then that transformed over however many hundreds of thousands to millions of years into a human, and there was no species left behind in that particular line of evolution, whereas cladogenesis is the divergence of an ancestral species. So usually we consider this to be known as a common ancestor into two sister species or taxa. And in this one, and I think the diagram in the next page will illustrate this much easier, so you have an idea of what the heck I'm even talking about. But in this case, there are branches to this ancestral tree, starting with a common ancestor, and then you have this kind of ancestral line with these extra branches off of it for each new species. And so this produces species diversity, oftentimes seen in fossil records and things of that nature. Now, modern day systematics and taxonomy and evolutionary biologists recognize cladogenesis as the kind of true way of looking at this. It's the only one that's really quantifiably accurate, the only one where you actually can have data that supports it through the fossil record. So anagenesis is very hard to quantify. As a result, it's the one that kind of gets pushed to the wayside, and most of the people who deal in ancestral origin of species kind of dabble in this cladogenesis. And ultimately they lead to the same thing. So if we see anagenesis here on the left side, it leads to this idea of kind of an ape-like ancestor turning into a human. But in cladogenesis, you get the same thing. You'd have some kind of ape-like common ancestor that ultimately leads to modern-day humans. But along the way, you have these separate species that are seen and maybe even lived at the same time or close to the same time as humans. And so some people think like Homo erectus and Homo sapiens may have occupied similar spaces in similar times as a result, whereas in anagenesis that would not be possible. It says Homo erectus effectively turned into Homo sapiens. So cladogenesis is really the one that we're going to go with, and in chapter 25 actually we build these cladograms. Okay, so we don't build these anagrams, we build cladograms. So it's the one that science kind of latches onto as the more correct way of identifying how species were formed. All right, so let's jump right in here. And one of the first things we have to understand in the origin of new species is the definition of a species. And so sometimes that can be complicated. There are multiple definitions of species, but we are going to use something known as the biological species concept. Okay? The biological species concept emphasizes something called reproductive isolation, where if you have two different species, then they shouldn't be able to reproduce and make viable offspring. If they are of the same species, they should be able to, mate with each other and produce viable offspring. So a good example of this would be a biological species, by this definition, would be like Canis familiaris, dogs, right? You can have a golden retriever and a golden lab mate, right? They are both dogs. They look different, but they are still of the same species, because if you mate them together, they can still interbreed and produce viable, fertile offspring. It might be a mutt, what we call that mixture. It might be a new breed, but theoretically that baby dog that is a result of that breeding could have babies of its own. And that's how you know the two parents were still technically part of biological species. So we're going to delve into this idea of something known as reproductive isolation, because reproductive isolation effectively defines whether or not two organisms are species or not. Reproductive isolation is the existence of some kind of biological factor. Oftentimes it's called a barrier, which means it gets in the way of these two organisms producing viable, fertile offspring. And that's the key. If we're looking at two species and they mate together, if they produce an offspring that can then have babies of its own, then those two original parents were of the same species. Now, these reproductive isolation barriers get in the way of two possible species mating with one another. And there are two different types of barriers. They are called prezygotic and postzygotic. And I think we should probably at least break down those two words. So the term zygote means, if you want to try and pause this and answer that yourself, feel free to, but the term zygote illustrates to us that there has been a sperm and an egg that have combined. We call that that very first cell of the organism, we call it the zygote. The fertilized egg is called the zygote, okay? And so there are certain barriers that get in the way of this union actually happening. We call those pre-zygotic, before the zygote. And then there are other barriers that get in the way of producing viable, fertile offspring by post-zygotic mechanisms, okay? So happening after the zygote's formed. So post-zygotic mechanisms take the sperm, take the egg, there is fertilization, but something happens after fertilization that creates non-viable, non-fertile hybrids. All right. So let's tackle the prezygotic barriers first. Prezygotic barriers impede mating or hinder fertilization if mating does occur. So in this case, these things that happen before the zygote, right? Mating between one organism and another has to occur. Okay, the actual act of sexual intercourse with the organism has to happen. Interaction between the two organisms has to happen, and these barriers get in the way of those types of things. Okay, so if two different species share some of these isolation mechanisms, we can understand that they have been isolated and they are two different species from one another. So all of this is in an attempt to define what a species is and be able to identify whether or not two totally different organisms are different species or not. And if any of these barriers get in the way of them mating with one another and producing a viable fertile offspring, then we can say that those two organisms are in fact different species. Okay, so we're going to talk about habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. So I want to do this in the format of just using pictures really more than anything to discuss them. The first isolation barrier is called habitat isolation, and basically what this means is that two different species, although maybe closely related, are still different, inhabit different habitats. They live in different habitats, and as a result, if they live in different habitats, they would never get together to actually do the act of mating. So these little grasshopper-looking guys here, this one on the left here lives in a sandy soil. This one on the right prefers a loamy soil. And as a result, they're going to hang out in their respective habitats and just never come together. So this is a pre-zygotic barrier, because if they live in two totally different habitats, they will never come together to do the act of mating, and as a result, they will never make a zygote, right? So this is a pre-zygotic barrier that keeps these two closely related species from mating. We have something known as temporal isolation, and for this I'm going to show these two slides and kind of bounce back and forth between them. They look like very similar organisms. I think they're both members of the skunk family. But temporal isolation refers to time, okay? And specifically these two reach sexual maturity and mate during different times of the year, okay? This one mates in late winter. This one mates in late summer. So in this case, if their bodies aren't even ready for the act of mating, like there are certain physiological things that have to happen in the bodies of many different types of organisms, especially mammals, where maybe the female has to be ovulating and be in what's called estrus or heat. And that only happens during certain times of the year for certain animals. And if they are not in heat or in estrus or ovulating, they physically, literally cannot get pregnant. And so this act of estrus or being in heat for this particular organism here happens in late winter, whereas this one happens in late summer. The sexual receptivity of these two different organisms at different times of the year is going to get in the way of those two organisms producing a zygote, right? So this isolative barrier takes place before the zygote happens, because if they're never ready to mate at the same time, then they're never going to mate, right? So there is a reproductive isolation barrier that creates the situation where they just never form zygotes, which is prezygotic. Behavioral isolation. We've seen mating dances of birds. We've seen the mating dance of the blue-footed booby before as well. And so you can think of two different types of organisms. So for instance, some of these birds of paradise that we've seen, they are of the bird class, the bird family if you want, the same as blue-footed boobies, but their mating dances are far different than the blue-footed booby mating dance, and those two things are behaviors. So if you take one of the birds of paradise that we saw in the video the other day, and you introduce it to a blue-footed booby, and they both do their respective mating dances, they're going to look at each other sideways and be like, what are you doing? That's stupid. That's a stupid dance. My dance is way better. And they would never mate. Okay, they would not meet the requisite dance steps, and both of them would kind of look at each other silly and just kind of walk the other way. And as a result, no mating happens, so that is a reproductive barrier, right? If no mating occurs, no zygote happens, it's prezygotic. Okay? Mechanical isolation can be looked at in two different ways. The example here is a little bit more abstract in the fact that these two flowers are of different species. They're closely related, but they have distinct appearances that attract different pollinators. So if this one happens to attract, this orange one happens to attract bees, right, and this purple one happens to attract hummingbirds, the bee will never, from this one, will never visit this one. And so there will never be any fertilization or zygote that's made because of the two. Another one's a little bit more risque. Another example is a little bit more risque when we talk about mechanical isolation. Sometimes parts just don't fit. I mean, this is a gross example and a gross thing to even visualize, but I mean, it is for biology class here, and it is a very good example of this. But think of an elephant, a male elephant, and think of maybe a female chihuahua. Right? The parts just don't fit right. And if that doesn't fit right, those zygotes never happen, they're never formed. And so these are examples of pre-zygotic barriers because they are things that get in the way of the zygote ever actually being formed. Okay? So try to wipe your mind of that gross example, but it should at least help you memorize what mechanical isolation is. Gametic isolation happens when gametes just don't fuse. So even though these sea urchins here look like they're probably closely related, we have a purple version and like a red version. Those two produce different gametes that for whatever reason just don't fuse with each other. So proper fusion of egg and sperm is required to create the zygote, right? So if you have two different types of gametes that just don't fuse with each other, you'll never get a zygote. So this is a barrier that gets in the way of, again, creating a zygote, so we call it pre-zygotic. Now, this I like this flow chart because it helps you visualize kind of what happens with these two different species. So if we've got a blue species and an orange species, shows that habitat isolation gets in the way of the mating attempt really before anything else. That's what we see here. The end goal would be a mating attempt. Whatever habitat isolation, they occupy different places. They're never going to mate, right? They occupy different estrus times of the year. Temporal isolation, they'll never mate. They'll just never come together to do the act of mating. And behavioral isolation, they have different weird dances that doesn't turn the other one on. They're never going to mate. Whereas we've got these other isolations, these other reproductive isolations like mechanical isolation, totally gets in the way of fertilization, right? That the process just might not happen. And we have gametic isolation, where the gametes just don't fuse. So this instance sometimes here describes what would happen in terms of if there was an attempt to mate. Okay, whereas here these three just totally get in the way of the mating attempt, and they cause a situation where the mating attempt would never even happen. Mechanical isolation, the mating attempt can at least be tried, so can gametic isolation, but those two isolations get in the way of actual fertilization. Okay. So let's talk about post-zygotic barriers. So let's pretend that the mating attempt happened, okay, number one, and the mating attempt was successful, meaning that fertilization of the zygote did happen. So we've got a zygote. These barriers help us understand what happens, how we can, how we can determine whether or not these two species are different from one another based on whether or not the fertilization attempt is successful. Okay, so these barriers prevent the hybrid zygote from developing into a viable fertile adult. Again, always keep in mind that to distinguish between two different species, we have to make sure that they don't produce viable fertile offspring. If they do, they are of the same species. Okay, so these post-zygotic barriers are listed here. There's only 3 of them this time as opposed to the 5 from before. But There are situations where the hybrid's viability can be reduced, okay? And in this case, when you hear the term viability, that should indicate to you how well the actual child or progeny or offspring can survive, okay? Reduced hybrid fertility gets in the way of the hybrid actually creating its own babies. So maybe I should have talked about that before. What does it mean to have a viable, fertile offspring? Viable means it's alive and will live to reproductive age, and fertile means it can reproduce, that it can make its own babies, okay? So we have different barriers that interrupt how well the organism, the offspring, survives, and we have another barrier that gets in the way of how well the offspring can reproduce, okay? There's a third type called hybrid breakdown, where you see some first-generation hybrids are very, very fertile, and they're actually really, really robust and very viable. But when they mate with another species, it's like this second generation is very feeble or sometimes even sterile. So it's almost like delayed a generation. So the salamander is an example of reduced hybrid viability. This particular breed of salamander, if you read in a book, is an example of two maybe different salamander species that came together and mated, and this guy is just not up to the challenge of living. So very rarely, if at all, does this thing actually live to reproductive age, either because it has birth defects that cause it to die really early, or it's just not able to survive for whatever reason. It never lives past reproductive age. Okay? Reduced hybrid fertility is one that we've probably seen many examples of here with the mule. Okay, a horse and a donkey are technically they're related. Okay, they're related species, but they are technically different species. They can mate with each other, and they can even have a baby, and that baby is called a mule. But the mule itself can't reproduce. It's actually sterile. You will never have any baby mules running around. You can have baby donkeys, you can have baby horses, but when a horse and a donkey mate, they can form a baby, so they get past those prezygotic barriers, but they run into these postzygotic barriers, which decrease the hybrid, which is the mule, decrease the hybrid's fertility to the point where it can't reproduce at all. It's sterile, meaning it won't make its own gametes. Hybrid breakdown illustrated here. You have like this plant on the left here is maybe the parental generation of one species and mates with another species, and it produces this big one on the right. It's very robust, it's very viable, and it's very fertile. But when this one goes to reproduce, it produces this piddly little thing, which is not going to reproduce hardly at all, or would be washed out in a very swift rain, a very harsh rainstorm, or blown away in a windstorm or something like that. It's just not viable enough to make it to even reproductive age. So hybrid breakdown is kind of like delayed hybrid viability and hybrid fertility all kind of rolled up in one. You get a really good first generation hybrid, but the second generation hybrid is just kind of crap. Okay? And so this flow chart kind of does the same thing as the previous one. Talks about, okay, fertilization has happened, reduced hybrid viability, shuts down a viable fertile offspring, reduced hybrid viability, or fertility shuts this thing down as well. And if all of those things happen to be bypassed, then you have a viable fertile offspring, and then the two parents that did the active mating are in fact of the same species. So that's how we define these biological species. We define them by whether or not they can produce viable fertile offspring. And we can determine whether or not they produce viable fertile offspring by examining the either prezygotic and postzygotic barriers. In the next section, I'm going to talk about speciation, how we actually get these different species. Now, no longer do we need to define them, but we're going to talk about how they are actually produced, okay? So I will see you shortly with that one.